Society on a lovely Friday afternoon. My name is Roshan and I'll be chairing the talk this evening. Before I introduce tonight's speaker, I'd like to take a moment to provide a brief introduction to OUPM for any newcomers. So the Oxford Personalised Medicine Society is a community of students and researchers from around Oxford University with the central aim of encouraging interdisciplinary research into personalised medicine and fostering discussion about its impact on healthcare. Now, personalised medicine is a buzzword which is used a lot, but its meaning is often misunderstood. Put simply, clinicians and scientists are capable of gathering more diagnostic and genomic data about their patients than ever before. Personalised medicine is a movement based around using this data to improve prediction and prevention of disease, to make diagnosis more precise, to personalise interventions, and all with the aim of increasing patient involvement in their own care. Tonight, I'm honoured to introduce Professor Sir Mark Caulfield, who will be talking to us today about transforming genomic healthcare. Now, Professor Sir Mark Caulfield, has an amazing CV, but I'm going to try and summarise it very briefly. He is currently the Chief Scientist for Genomics England from 2013 till present. He oversaw the delivery of the 100,000 Genomes Project. He was the director of the William Harvey Research Institute, president of the British Hypertension Society, and is now professor of clinical pharmacology at the William Harvey Research Institute, among many other things. Since 2014, he has been one of the top 200 most highly cited genomics researchers in the world. We are very honored to have Sir Mark Caulfield talk to us today. Now there will be a Q&A session at the end. To submit questions, please could you message them directly to the Centre for Personalised Medicine account in the chat, and we'll have 15 or 20 minutes at the end to talk with Mark. Thank you very much. I'll pass it over to you, and you can start sharing your screen.
So it's a real pleasure to be here, and thank you, Roshan, and all of this society for inviting me. It's a particular privilege to be doing this in Oxford, although I wish I was with you. I'm mildly surprised that you want me to do this evening at this time, because I might have expected you to be in another place at that time, but there we are. But it's a testament to you, and all I can say is that you're in absolutely the right place, because genomic medicine is here and in the clinic, and it's really, really a pleasure to see so many of you here. So I'm going to talk about transforming genomic healthcare. If my internet, which is provided by Sir Richard Branson, and therefore is entirely flaky, cuts out, Roshan's going to tell me, and I'll go back and just cover the point again. So these are my disclosures. I don't really get any money other than my salary from anybody these days, because essentially I work for you, the government, and taxpayer in the 100,000 Genomes Project. So the 100,000 Genomes Project is a really unusual origin. It started here. This is the London 2012 Olympics, and that location in 2012, the then Prime Minister David Cameron gathered health ministers and experts from around the world, and they had a summit. And the experts in the field of genomics suggested that it would be the right moment for a free at point of care health system like the NHS to consider whole genome sequencing. And the 100,000 Genomes program was born. Now, for some of you, this is really very basic, but just to remind those of you who are coming to this because you're interested but don't know quite as much as everybody else, your genome's about 3.3 billion letters, so you're a billionaire. And you spend those letters making proteins and regulating essentially your genome. And probably every 300 letters or so, you've got a variation. They can take different formats. They can be single letter changes, a single nucleotide variant. They can be extra bits or missing bits of DNA, or they can be copied bits of DNA, and there can be structural connections. When Professor Todd and I were younger, we used to work with a type of variant called microsatellites, which we don't work with quite so much now. And then we moved on to large-scale genotyping once the initial human genome sequence was produced. And John and I were to work together in the Wellcome Trust Case Control Consortium in 2007, which was led from Oxford. And actually, was really the first major collaborative study in genomics in the world, and it ushered in a whole era of collaboration. But now we've moved somewhat to next generation sequencing, and we can read, of course, as you know, which is the 1% of your and my genome that codes for the proteins. We can also read the whole genome, or as much of it as we can read today. And through integrating that with advanced bioinformatics, we can begin to make this tractable to disease. So what was the 100,000 Genomes Project? Well, it was looking at whole genomes, and advisory groups suggested this focused on rare disease, cancer, and infection. And the reasons were because it's tractable to benefit in the healthcare, or was in 2013 when we started. The program's reach now over 97,000 people affected either by rare disease or cancer from families or individuals. And we actually have much more data than 21 petabytes now. We have over 40 petabytes of data. So we store that in a large data center. We're increasingly migrating it to the cloud, but it is stored in a fixed data center, which is not too far from you in Oxford. It's in Corsham in Wiltshire, in a big government data center there. To do this, we had to create infrastructure in the NHS. So what I recognized and had known for some time is there were many excellent colleagues working extremely hard with very limited resources, was to galvanize those individuals together and give them a banner to unite around in a region. So just as there was in Oxford, there were 13 genomic medicine centres in total across England, and Scotland, Northern Ireland and Wales joined in, and that was very important to me. Out of 220 NHS trusts, we reached 98 hospitals, and at peak, 5,000 frontline NHS staff worked on this programme. That's really important because if you or I want to embed something in healthcare, we need to involve the people at the coalface who do this. And the 5,000 people would touch the project at some point in their week, and they would all be part of the multidisciplinary healthcare team. What that meant is by the end of the project, we had a cadre of people who were willing to champion the inception of this indirect healthcare. And therefore, we short-circuited the usual median of 9 to 16 years from a clinical research program publishing to adoption in the health system. Now, because this technology and how we deploy it in the health system isn't fully mature, we needed to establish a coalition of research intellects, and we did that by open advert in Nature a couple of times. And any of you here can, for free, access our data. You just have to be in the Genomics England Clinical Interpretation Partnership. And there's now 3,580 researchers, I believe, who have access to data. And I'll show you a little bit about what they've done with that. So just to give you a sense of the infrastructure we had to build, so in 2013, genomic healthcare in the NHS was a bit of a postcode lottery. So there were some parts of the country where you could get pretty good testing, not whole genomes, but you could get exomes, you could get panels, and you could get access to tests. And there were other parts where effectively just a few miles down the road, you would get limited testing, if any. And so this had to democratize access to genomics and create a platform for equity of access. So the Genomic Medicine created regional equity of access to the 100,000 Genomes Project. And Scotland, Northern Ireland, and Wales joined in too. Northern Ireland and Wales had their own genomic medicine centre, and Scotland did something slightly different, but that was fine because they were in the programme. So the frontline NHS teams consented the patients to be involved and took the samples. And those samples were sent to a central biorepository, which is the UK Biocentre in Milton Keynes. They also submitted clinical data, and this was really, really important because particularly in rare disease, the clinical data makes all the difference to making a diagnosis. And we also then collect every electronic piece of health data we can find on all the participants, and we follow them for their entire life course, including until after their death, so that we can build up a granular picture of these people and their journey through their problems. And that may help us inform the biology and also the therapy of the disease downstream. So we created a sequencing centre. So we got £27 million from the Wellcome Trust given to the Sanger to create as a football pitch-sized sequencing center. And we got Illumina to run that sequencing center, and that proved to be a very good partnership because we got access to the best price worldwide, the latest chemistry and the latest technology. And Illumina, who own the technology, took the risk of running it. So we built a Genomics England informatics architecture. This is where you would come and do your research. So it's a reading library, not a lending library. You could come in as I'm speaking to you now over the internet through your laptop and access the research data we have. And that's through the Genomics England Clinical Interpretation Partnership, or GSIP. And whether you're a student, undergrad, postgrad, whatever you are, you're more than welcome. Just apply online, go to GSIP at Genomics England, and it will show you how to do that. A discovery forum is a coalition of companies who we work with to try and drive up value of the data. And the whole goal of both the academic research endeavor and the industry is to drive up value for patients and get new therapies. And then, of course, the distinction between standard research projects in this is we return a report to the NHS, to the clinical teams, and they own the relationship with the patient, and they validate and return the findings if there's something to say. So here's the journey that a genome goes on. So this DNA molecule at the top is taken from patient or family that have consented, and then it becomes DNA, is sequenced. By the way, no genome moves as fast as this one through the system. The genome sequence is annotated. So we annotate that to allow us to tier the variants that could be relevant for the disease, either in cancer or in rare disease or infection. And then we review that. We had some help from some annotation companies. We return it for clinical assessment in the NHS in a standardized report. And at the same time, it goes off into the red box for research done by people like you guys. So I'm going to focus on rare disease for a bit. Rare diseases, although they're individually rare, there's over 8,000 of them, and some people believe 10,000. And I suspect that will rise until we really have identified every rare disease. And we may never do that because sometimes new mutations crop up spontaneously at conception, and these can cause completely new diseases we've never seen before. We worked on 1,200 disorders with unmet needs. So the whole premise of this program was you had to have no diagnosis yet. And just to share with you that of the people with rare disease, because it affects 6% of the UK population, it's 1 in 17 people. So that's because there's so many rare diseases, although they are individually rare. But if I share with you that when we started, approximately half of the people would go through life without a genomic diagnosis. They might know that they had a syndrome of one sort or another, but they wouldn't necessarily get a molecular diagnosis. Really key to success was standardizing eligibility and phenotyping. So we use something called the human phenotyping ontology. What that really is a way of making my clinical terms standardized so that if I need to share data with another country or another place or another hospital, I can do so. And because I standardize that data, we're instead of using apples and pears to speak to each other, which are all the same fruit, we can all speak in apples, and therefore our data can be combined and usefully yield insights into diagnosis. In a moment, I'll show you how important that was. We also developed semi-automated analytics. And when the NHS were invited in, they confirmed the gene panels we initially looked at. And again, eventually we went genome wide, and they also own the closing of the cases and the return of reports. You can see on the disorders that we've worked on sit by different systems at the bottom. And at the top right, you can see the first family who received a result. So the little girl in the middle was 4 then, she's 9 now. And she had developed a developmental delay at 4 months. She was falling behind her peers. And then she developed intractable seizures. So in the time we're here today, she would have had a fit. So she had a mutation in her glucose transporter gene, which we found. Her mum and dad had enrolled her in multiple genetic projects before, but no diagnosis had been made. This was a very important discovery because it paved the way in her case for a high-fat diet, a ketogenic diet. And because of a starvation mechanism, you and I can make sugar in our brain from fat, and that's what she did. And now her seizures have really subsided, which wouldn't respond to any anti-epileptic medicine. And she's had some developmental improvement. What if we could have caught this at birth? Interestingly, also, this is a family-wide event. So mum and dad had not been going to have any further children, but because they discovered this was de novo in their daughter, in other words, it had occurred at conception, they are since children. So let me take you on the journey of her genome. So I shared with you it's 3.3 billion letters, roughly in total. In her case, there were 6.4 million variants, 677,000 were rare, 2,826 changed a protein, 67 are different from her parents, and 1 is a clear-cut cause of her glucose transporter 1 deficiency. So that's how we find it. And we do that in a few hours in parallel for families, and we analyze the families together because it improves line of sight on the diagnosis. Let me give you some other cases. The first one's a sad story. It's a 4-month-old child who was born, went straight to the neonatal intensive care and never left it. He died there with no diagnosis. Immune testing was negative, and his parents enrolled him in the project. His mum became unexpectedly pregnant, and we found a pathogenic transcobalamin 2 mutation. His brother was born and tested and sadly was affected. But because of case reports, in our report, we were able to say that some children had received weekly doses of high dose vitamin B12, and that had ameliorated symptoms and that they'd had a better life course. And so that was done. And this child has some minor intellectual delay, but other than that, he's at a completely different life course and is still alive today. This 10-year-old girl has been admitted recurrently to hospital with life-threatening infections, on this occasion chickenpox. Detailed immune testing had been negative. We found mutations in the CTP synthase 1 gene, which affects B and T lymphocyte responses to infection of both bacteria and viruses. She has had a curative bone marrow transplant. Her siblings have been tested and they're not at risk of these infections. So let me give you an idea now of some economics here. Now, you might think doing a whole genome is very expensive. I share with you today it's less than $600 to do a whole genome in a rare disease, and that the course of this child had cost the NHS £356,000, and for £70,000 she had a curative bone marrow transplant. What if we could have deployed a whole genome at the first sign of trouble, made the diagnosis much earlier, effected an expensive treatment, but far less expensive than presiding over a diagnostic odyssey of 10 years, where we don't have a diagnosis and we spend £356,000 instead. The final one is an example of how the genome can reprofile a diagnosis. So this child, a 5-year-old, had unexplained anemia, developmental delay, a short stature and constipation. Given a diagnosis of Diamond-Blackfan anemia, which can have a limited lifespan due to cancer, because of his intellectual disability, he was examined by the intellectual disability, his genome was tested by the intellectual disability panel, and we found that this child, instead of having Diamond-Blackfan anemia, actually had a thyroid hormone receptor alpha mutation. That's a known pathogenic mutation that just had never been looked for because no one had suspected it. And he's now receiving high-dose thyroxine. And you can see a video of this child on our website. I didn't dare put it in here because I didn't think it would work well on the internet, but you're welcome to go and look at it. And what you see in the early part of the video is not long after the diagnosis. And then towards the end, you see a little bit of a follow-up bit, and you can see at the beginning, he's struggling to walk properly on his own. By the end, he's dancing. Now, he will never be entirely normal, but what if, again, we had found this earlier in his life? So this is the perspective of a mother in the program. I'm going to read this because this is her words, not mine. And I'll just summarize the case while you're reading. Her child was 5. She'd been diagnosed with GAMP deficiency, and we found that she had the mutation for GAMP deficiency, which was a treatable metabolic disorder. Now, no one had really known what she had, but our genome revealed that she had GAMP deficiency. And before that, she was on the course to a degenerative neurological disorder, potentially ending in a wheelchair, if not worse, uncontrollable epilepsy, multiple fits every day, a development age of 12 months, and becoming increasingly locked in. After 6 months of simple treatment with something you and I have eaten in the last 24 hours, creatine, ornithine, and arginine, she's a new child with the light back in her eyes. and she can communicate now. She will never be normal, but it's far better. Now, to you, this is going to happen for every child with a rare disease, but I am saying we have the door is now open for some of this. What if we put a platform of therapeutic innovation alongside this? Sorry about that noise, I'll get rid of it. And what if we did that together? What could we achieve for these children in the future? And the beginnings of rare disease treatments now. So for some disorders, they're becoming more amenable to therapy. I'm just going to show you some data now. So we've got a paper hopefully about to be accepted at the New England Journal of Medicine, though they have given us a bit of a hard time with it, on 2,183 families with 160 rare disease categories. And so that reaches 4,660 people. What I want to draw to your attention is that we achieved a bit over a 25% diagnostic yield. And you'll see that diagnostic yield is proportional to the family size. So if you're one affected individual, you're less likely to get a diagnosis than if you're a duo, a mum and usually a mum or a dad and a child, or a trio, which is two parents and one affected individual. So line of sight on a diagnosis is increased by increasing family number. On the right-hand side, you can see that we achieved about a 37% diagnosis in likely monogenic diseases. We did allow some more complex etiology diseases that might have an oligogenic, a few gene architecture. And then on the right in the pie chart, what you can see is the blue is what you might have found from a standard panel or an exome. And in the yellow, green, and orange, that's what you get from a genome. So there's huge added value of a genome in this approach. So just to share with you what the diagnostic yield is, remember that every single person in this program has not had a diagnosis, and some of them have had prior genetic testing that's quite extensive. I've set the bar here at 25% uplift, so you can see some disorders, hearing and sight, metabolic disease, intellectual disability, neurodevelopmental disorders, ophthalmological, all have increased above 25%. But there's reasonably appreciable uplift elsewhere. Let's take a look at what the impact of this technology is. So this is something I didn't think we'd find. What you're looking at here are disorders that are caused by what were called simple tandem repeats. Some people call these dynamic polymorphisms because they expand in succeeding generations. So to the left of the red dotted line, what you're seeing is the normal profile from the totality of the genomes that we have of repeat distributions. So these are repetitive polymorphisms. If these repeats are expanded to the right of the red dotted line, then that's a repeat expansion, and that can cause disease. So the top three are hereditary ataxias caused by different genes. In the dark red are substantial repeat expansions. Now, we didn't think a short read technology would allow us to see that, but using a program called Expansion Hunter, which is an informatics program, we can measure these things and make these diagnoses. And we have a paper in advanced review at Lancet Neurology, which is describing this and its validation using orthogonal techniques. I just want to draw your attention to the bottom row. So in the bottom row, this is Huntington's disease. And to the right, you see two red triangles that are shaded. Those red triangles are children. And one of them is 10 years old and are presented with ataxia, intellectual disability, an atypical presentation in children, and not one that people would normally have made a diagnosis at that age of Huntington's disease, usually a disorder diagnosed between 20 and 40. But this child has Huntington's disease. Now, that might previously have been a really disastrous diagnosis and still is for many people. There is some hope in that there is now an antisense oligonucleotide which knocks down the deleterious overexpression at the Huntington's locus, and that actually can ameliorate some symptoms and signs. And as some of you will know, once you've one therapy, you can potentially get others. This is the diagnostic uplift across the pilot cases, which is roughly around 1/3 for about 33% for whether you had no prior testing or prior testing. So you can see that there's a dramatic uplift. And on the right-hand side, it's only a few cases. So these are the same pilot cases who didn't achieve a diagnosis. Some of them had multiple tests. So there's one person here with 16 different tests. And this takes a long time, because what happens is they come and have one test, then they go away for 3 months while they analyze the test, then they come back, they have another test, and you can see how that builds to years. So from that pilot, 24% had an immediate clinical use. You can see here what they got. Some got access to a trial, some got access to diet, some got medication shifts, some got additional surveillance. There were, by gene burden testing, over 500 genome-wide significant signals for further functional exploration. So the diagnostic yield is not yet finalized. We have to do further functional studies, and that often slows us down. Let's take a look at the diagnostic odyssey of the children in this pilot born after 2003. So really, this will possibly surprise some of you, although others will be familiar with it. Families spent 6 years attending a median of 68 hospital appointments, compared to their siblings from the same family who attended a median of 18 hospital appointments over 120 months from birth. After diagnosis, we see a fewer number of focused clinical episodes. These affected participants used 183,273 episodes of care at a cost of £87 million. So that makes a $600 genome look kind of good value at £15,000 per participant, compared to 53,000 episodes in their family members at a cost of £21 million. That doesn't include visits to the family physician, which is GP in this country, or direct treatment costs. So this is the data on the full main program. We've analyzed over 36,000 families here, with 6,613 different phenotypes from about 1,200 disorders. And the current diagnostic yield, though we're doing additional research on that all the time, is 20%. So it's slightly lower than in the pilot, but we did a lot of extra research. So the job is not yet done, and that's why I need you to come and turn up and help drive up the value of this data in the clinic by joining our research coalition. Going to focus in a bit on cancer now. Cancer is by definition a disease of disordered genomes. And this proved really challenging, because the standard preparation by which we use for cancer is to drop the tissue into formalin. This is very damaging to nucleic acid. And here you see a circus plot where we've arranged the genome round in a circle from chromosome 1 to X. And what you're seeing here in the middle, in the center of the genome, is structural connections. And what you can see is prostate cancer from the same man's prostate cancer biopsied on the left and right. One went down a fresh tissue route, that's the left-hand one. The other one on the right went down a formalin fixed route. And what you can see is the structural connections on the left are far less. There are far less S single nucleotide variants or insertion deletions. And therefore on the right, what you see is a more moth-eaten genome. If you look at the outer layers, you can see it's uneven coverage. And then in the middle, you see it looks like a child has scribbled on the genome. The product on the left is usable in the health system. The product on the right is not usable in the health system. So we re-engineered 400 molecular tissue pathways across the NHS in England, and that allowed us to enroll 17,400 plus people across all these cancers. This is the largest whole genome cancer resource in the world, and it's there for you to work on. And you can see that it's dominated by common cancers, but some people put particular effort into gaining access to technologies such as sarcoma with 1,652 patients. Here I'm looking at mutational burden, and I'm looking at it by tumor type. So we're moving by tumor type across on the bottom, and up the side it's tumor mutational burden per megabase of coding sequence. And in the serrated box there, you can see where the line is drawn where the tumor mutational burden is high. Why is this important? Well, high tumor mutational burden may suggest that you should be prioritized for earlier immunotherapies in cancer. Now we have therapies that we can target using this. If you particularly look at, say, endometrial cancer or colon cancer, you can see the higher mutational burden, and almost clear sky between that and a lower mutational burden. Here's a family to illustrate the impact on individuals. So in the family tree, squares are men, circles are women. The diagonal line means they're deceased. And the shaded circle here is an affected individual that actually is from a family that's got a negative history of breast cancer. But this woman had breast cancer, and she had BRCA2 in both her tumor and germline genome. And that paved the way for her to enter the Olympia trial with the olaparib. And her daughter was also tested and turned out to be BRCA2 positive with a pathogenic mutation. And she, as a result, has entered yearly breast screening with scans, MRI scans. And her male family members on the right-hand side are also considering BRCA2 testing because it can be linked to prostate cancer. So this is to emphasize to you the familial wide impact of this result. Let me show you another case here. This is a man who was 42 and had a previous hepatitis B infection. And what you're seeing here is a CT scan on the right-hand side. This is a slice through the center of the person's upper abdomen, and it's cutting through the liver. And you can see a grayer dense area in the liver. That is colon cancer that has metastasized to the liver. That was biopsied and had usual care testing, which showed no mutations that changed therapy. Because of progression and increasing symptomatology in his youth, we did a rebiopsy, did a whole genome sequence, and this man turned out to have hereditary predisposition to cancer with Lynch syndrome, having a microsatellite mismatch repair gene. And as a result, this man entered an immunotherapy trial. Now, he didn't survive, but perhaps he had better quality of life as a result of that. What if we could have discovered this earlier because we knew his hereditary predisposition by surveying his colon on an annual basis to detect new tumors, and then this could have been treatable. So the big question here is, what did the NHS do with these cancer reports? Because there wasn't much molecular testing for cancer when we started. Well, this is the data from the West Midlands Genomic Medicine Centre, and every centre had to have a genomic. So of 3,244 patients who were entered in West Midlands, 781 as a result of their genome. Here we read the genome of the tumour and compare it to the genome you inherit from your mum and dad, usually in the bloodstream. And by subtracting your inherited genome from the tumour genome, we get the drivers to the cancer. And 781 people had a finding in their tumour genome that suggests the Tumour Advisory Board should look at it again. And 477, that could include detection of a pharmacogenetic polymorphism, DPYD, that suggests you should have reduced dose or not have 5-fluorouracil or capecitabine. It changed for 21 the clinical licensed drug that they had. For 11, it changed an unlicensed to an unlicensed drug, 204 into a clinical trial, 43 of high tumor mutational burden and therefore would be amenable to potential immunotherapy, and 69 were referred to clinical genetics. So approximately 25% of the patients went back to the tumor board, and some of those had a clear action in the clinic. And this will only increase in time. Let me draw your attention to the ones that were excluded, the 14, 16 in the middle box, just below the 781. Those people may not have an immediate benefit from their genome, but it's banked. So if they have a tumor recurrence, it's use. We also use this, and you may be interested in this, to help enrolment into the Focus 4 trial, an Oxford-based trial led by Professor Tim Morn in Oxford. And in the blue and the red are potentially eligible patients who we've identified by their genome. And we can't tell the clinician running the trial who the patients are, but we can tell the clinician who looks after the patients that they're eligible for this trial. And we can also tell the investigator running the trial, in this case, Tim Morn, where the patients are. So we can tell him that there's 15 people in Wessex and Dr. X is looking after all of them. So that's something we do in every report. We connect every cancer report to a clinical trial where we can. So we did some work in infection, and this is with Derek Crook again in Oxford, where we sequenced a total of 10,000 TB strains worldwide, 3,000 from the UK of multi-drug resistant. And this allowed us to create the evidence that you could diagnose TB effectively, but also that you could resistance profile within a further 24 hours of diagnosis. So get the right TB therapies to the patient. And that led in 2016 to a new TB service in PHE, where they sequence more than 1,000 organisms a month for resistance and also diagnosis. In pharmacogenetics, we analyzed how many of the Clinical Pharmacogenetics International Consortium variants are possessed. And we looked at 60,000 whole genomes. We've increased that now to 76,000 whole genomes. And I can tell you that 99.5% of us possess a gene-drug pair will, if we come across the therapy, either cause it to be ineffective or may cause us harm. So the NHS is getting ready to report these from whole genomes, but also to run panels that look at these in the NHS, so that we'll be able to use both preemptive genomics and responsive genomics. Now, if I also share with you that 6.5% of admissions in the English NHS are due to adverse reactions, they're not all due to pharmacogenomics. But what if we could bring this live? What if you carried around on your mobile phone a traffic light app that showed you red when doctor wants to prescribe a drug, you just plug the drug name in. It's red for don't have this drug, your genome is going to work well with the drug. Orange is you can have this medicine, but you either need to reduce the dose or increase the dose. And green is your genome and this drug are not going to be harmful if paired together, but you may still get side effects. What if we could achieve that? So now I'm going to share a little bit about you, where you come in. These are the 3,580 researchers from 33 institutions in, 413 institutions in 33 countries. They've raised over £50 million in research investment. That goes to them. And you can see the map of the world for where they are. In addition, we work with 130 companies. They have access to 3.8 billion clinical data points on 111,000 genomes, sitting there waiting for you to use it. We collect every piece of data we can, so it's a very rich data set, and we do periodic releases to upgrade the data so it's more usable. To be honest, initially it wasn't very good, but it's now getting much better. So have we used this data to transform healthcare? Well, in 2018, my team and I worked with the NHS to look at 300,000 tests used in the health system, upgraded 5% to new technologies, and made the case for 500,000 whole genomes available to clinicians for direct requests against specific disorders of rare disease and cancer. And the patient offered a consent for research, and that includes recall for research, longitudinal life course follow-up, and the availability of that data to UK and international researchers from academia and industry. And so the NHS then, alongside that, created a test directory from that work, and they used that to underpin a new NHS genomic medicine service with 7 NHS genomic lab hubs. And when patients consent, they will go in the new UK genomic knowledge base, or the National Genomic Research Library, as we now call it. Genomics England will continue to provide whole genome sequencing and as interpretation, and there are now 7 genomic medicine service alliances, which are regional collaboratives of hospitals to embed this in healthcare, and that will also lead to workforce expansion and training. But who would have thought there'd be 500,000 whole genomes, and for the first time, regional equity of access to genomics in the UK? I'm just going to home in before finishing up on severe COVID. So this is work we've done with a genomic study, and this is UK wide. We've worked with it's now up to 212 ITUs, 5,019 ITU beds, and we enrolled 2,224 severely ill COVID patients from ITU with Kenny Bailey in Edinburgh. And this also had colleagues like Julian Knight in Oxford involved. And what we did was to do genotyping chips, so not a whole genome here. They are being whole genome sequenced at the moment, but this was published because we wanted to get on with it quickly in Nature in December 2020. You can see 30% were women, the median age was 57. Sadly, 22% have died, but note this, 25% were from ethnic minorities. And here is what's called a Manhattan plot. So this is chromosome 1 across to X at the bottom, chromosomes, and what you're looking for is skyscrapers of variants exceeding the red dotted line across the middle of the picture, and that is achieving genome-wide significance with the log p-value up the side. And you can see a hit on chromosome 3 and others across there that harbor loci. So just to summarize that, we found 7 genome-wide significant loci, 3 identified potential therapies. Interferon 2 receptor is a potential target for interferon, obviously. That had been trialed and not been successful in severe ileal patients, but could be tried earlier. We also identified the tyrosine kinase 2 locus, the target for JAK inhibitors used in not cancer, but in conditions. That's an error. And one such, baricitinib, has now been adopted by the Oxford lead investigators, Peter Harvey and Martin Landry, in the recovery trial. And there's another region which might enable repurposing of a biologic tested in rheumatoid arthritis, because the locus on chromosome 3 we think is most interesting is chemokine receptor 2, which causes migration of monocytes to the site of inflammation. So we put public and patients at the heart of this program. And if you want to keep programs safe, you need to involve the people the program's about, so they own it and shape it, tell you when they don't like what you're doing, but also guide you and also provide you with support when dealing with people who don't think you should concentrate data. So we asked a representative group of the population across the UK what they thought were the key features in the NHS social contract that patients should adopt, and what were their red lines in the advent of a new genomic medicine service. So they identified reciprocity, altruism, and solidarity. They also said that they're not wild at the moment about gene editing in humans. They'd like to know more and see more data. They raised the need for us to use the data for commercial uses, but they don't want their data used for marketing. And they don't want healthcare data used for a surveillance society. And I can tell you that in 2013, I made sure that Genomics England has as an unacceptable use of its data for surveillance or forensic purposes, because that's a route to have non-engagement from certain sectors of our community, and therefore that's an unacceptable use. So what is the future program from Genomics England? Well, my time as chief scientist is drawing to a close, because in July 2021, I will stand aside because this is the right moment for someone else to pick up the relay baton, because the job isn't quite done. We have a new genomic strategy that's quite detailed, produced by the government. What I would say is I'm not showing you any pictures of politicians, because I suspect you're wild about seeing them. But whether you are or not, they have been steadfast supporters, and the current government have really got behind genomics. So I think they've done some good. The next program will address diversity and inclusion in genomics, because we're underrepresented for our communities that represent the full nation. And also there's an opportunity for us in Britain to act as a research window for other communities around the world where genomics is hard to do. And in particular there, we'll focus on pharmacogenomics. We'll have a Cancer 2.0 program. The one I'm particularly pleased about is the ability to look at diagnosing early newborn children, like the cases that I showed you. And we've looked at this quite carefully. There are about 600 conditions, 1 in 190 live births, 9 children born every day in England, who have a disorder that's treatable within the first 5 years of life. In other words, there is a therapy. Only 8% of these therapies are expensive. The majority are simple dietary changes, vitamin changes, and some are medicines, but most of them are not expensive. What if we could change this and usher in an era of genomic prevention and a platform for clinical trials that would encourage people to see Britain as the place to bring their new therapies for rare diseases to first? And we will also continue to work with the NHS on expansion of whole genome sequencing. I'll finish. John Todd and I were in the 100,000 Genomes Project. I know that John used to use my Manhattan plot as an example of a null experiment, because in 2007 we didn't find a sausage with the first Wellcome Trust Case Control Consortium. But because blood pressure is so important in stroke and heart attack and causes so many deaths, and 1.5 billion people will have hypertension by 2025, and blood pressure control is still a challenge, we kept going. And there were rare syndromic forms of blood pressure that were discovered. Can you hear me, Roshan?
Yes.
Good, okay, sorry, the internet's a bit flaky, I can tell that. But we kept going with blood pressure. And now I'm pleased to say Professor Todd can no longer my Manhattan plot as an example of a null experiment, because we've discovered over 1,200 loci for blood pressure. And in fact, I should give credit in this talk to my colleague Patricia Monroe and Professor Joe Houston from Oxford, who contribute significantly to these discoveries. This is a paper from 2018, if you're interested in Nature Genetics, that we published using a two-stage design. It's been a great boon for genomic research, where we discovered 535 novel loci. We also produced something that's quite topical now, which is a polygenic risk score, and that shows that across different deciles of the genotype relative risk score, you can see an increase of 13 millimeters of blood pressure in the over 50s, and an increasing risk with aggregate blood pressure variance as you increase the number of variants of cardiovascular disease of one sort or another. Interestingly, we've looked at in a clinical trial of about 8,000 people, and we find that the risk score also predicts resistance to hypertension treatment. So that's a new finding that we haven't yet published. I do believe, though, that whether it's rare disease, common disease, or whatever it is, we're in the era of multiomics and also single cell technologies. So I would encourage all of you to join this race by integrated multiomics, not just whole genomes. It's not one single thing. We also need transcriptomes, proteomes, metabolomes, and single cell omics with artificial intelligence. We still need experimental models. We need a national functional genomic strategy to lay a function on and give us the biology that we don't yet have to solve the findings that we can't yet explain. So, if genomically primed therapies are twice as likely to get into the clinic, then target validation for these findings must be our goal. We've used the blood pressure findings to incept studies in dietary nitrate, which shows it lowers blood pressure effectively in a 16-week trial. We've made new CNP, C-natriuretic peptide mimetics, which are being solubilized with 2 million from Apollo Therapeutics. And we've got adrenal adenoma therapies of a calcium channel blocker. And there's already genotype driven trials in high blood pressure, including adducin alpha and uromodulin, both of which are underway at the moment. And what you see here is the progression of discovery, and you see the increasing use of single stage design in blue, and in red, the emergence of rare variants, mostly from exome chips. But what this tells us, this is much more of the architecture of the traits we've been interested to find. And also, finally, that the effect size of rare variants is more appreciable than those of common variants, as we might have guessed. But it could be that rare variants will help us not just find MAP, but also refine subsets of populations that are distinctive, that could prime different therapeutic approaches. So I will summarize now to say, in 2021, the National Health Service has a genomic medicine service providing equitable access for, actually it's 56 million people, and Scotland, Northern Ireland and Wales will follow. It's got standardized protocols, standardized procedures, and as a single two-sided genomic consent for NHS care deployed across the whole of the population. So it's a uniform consent in every centre, delivering against the national test directory, building a single knowledge base for research, and providing they consent people, those will be available to you for research, are de-identified for academia and industry. It's been a real pleasure to join you this evening. I can't emphasize to you enough that genomics has paved the way and led the way in worldwide collaboration. And we must always, particularly at this time when we've chose to leave our partners in Europe, practice science without borders, because as COVID shows us, viruses don't know where the borders are, and we cannot defend ourselves against disease if we don't band together as a global coalition in genomics. Here are the most important people in the project, and they're counting on you to turn up and do research because they have not yet got answers, some of them. I'd also like to thank medical students and Students for Rare Disease Society. This is at my medical school, Barts in London at Queen Mary. And I'd like to thank you, Roshan and Holly, and the Oxford University Precision Medicine Society for inviting me to share with you this evening. Please do get in touch if you want to know more, and I'll stop there. Thank you so much.
Brilliant. Speaking on behalf of everyone, thank you so much, Sir Mark, for your amazing talk. We've got some questions from the audience, if you're happy to answer them.
Perfect. Press ahead.
So the first one from Professor Todd, actually. Is there any chance all the gel plasma samples could be analyzed using the latest deep targeted and untargeted proteomic technologies?
So, John, to answer that, and you might want to come back orally. First thing, I'm just doing, so I've identified the 6,000 that we have samples on, and I have sent them for high abundance mass spectrometry proteomics, and we've got a pilot of 500 underway with Olink and with SomaLogics. And for everybody, so proteins can be, this is too simplistic, but high abundance proteins can make it difficult to measure low abundance proteins. So there are affinity assays, either based on DNA aptamers or on antibodies that have been developed, and you can use those to measure, in the case of SomaScan, up to 7,000 features. So, John, we're doing that work with Markus Rauser in Berlin and Claudia Langenberg. Sorry, there's a bit of an echo there. I hope that answers that.
Yeah, thank you. The second question was, can you recall gel subjects for any genotype or any phenotype rather than only the condition that they were originally enrolled into gel?
No, so John, you can work on any condition or any exposure, because obviously, if they survive the rare disease, they may get other disorders. So basically, actually, I copied you, if I'm honest, because the recall for research was your idea in the bioresource. So I just copied that because it was obvious that in the future, we don't know what will happen to these people. And I tested it with the public, and they said, look, basically, if we could benefit from something, we'd love to know about it. So, we've practiced in the UK a habit of having too narrow a consent. We need a much broader consent to maximize the value of these patient engagements.
So, one last comment or question. Mark, that was superb. What next for Mark Caulfield?
John, what I'm doing, this is a natural break. So before, in 2019, actually, I think you were there, I consulted with scientists and industry, and we shaped this program. So we put it to the government last summer, and it's been funded for 1 year, and then we're going back in for a further 3 years funding. That's the newborns, diversity, and cancer. So I felt this was a natural break. Also, when you hold these jobs, you need to, at some point, to say it'd be good to have someone else do this and bring their ideas to it. So I felt it was time to get out because this was a natural break. So what I'm doing is two things. I'm going to go to the NHS to help with evidence generation, because what I've realized is you can push things at the NHS, but having created an annual review of the test directory to make sure it continues to be dynamic, I really, really wanted to go to the other side and pull stuff across. And unless they get some folk like you and I turning up over there and saying, look, this is okay, the inherent conservatism that often holds us back will not reflect the dynamism of this field. And if there's any message anyone can get from this talk this evening, genomic medicine is here. It's in the NHS. It's coming to most diseases. I can't guarantee all, but I suspect in the next 5 years, the role of genomics will become much more important. I hope that answers things for you, John.
Thank you.
Oh, thank you for your kind words.
Yeah, great to hear from Professor Todd as well. Lovely. Next one is from Max Cowan. My understanding is that the current Illumina technologies used in the Genomics England pipeline are short read sequences. Is there any vision to integrate long read technology?
Yes, absolutely. Spot on question. So short reads, what we do is we make a library prep where we fragment the genome into 150 base pair lengths, and then we reassemble it on a reference genome. That makes it difficult to read repetitive regions, and some regions we just don't get as good coverage. So we are entering a partnership, and we do have Oxford Nanopore technology. So from your neighborhood, that's a long read genome, which reads in gigabases, so much longer. And it reads by passing nucleic acid through a pore. So it reads as it goes through the pore. Now, the great thing about that is in cancer, you can get real-time readouts from the pore. So for acute leukemias, where the doubling time of the tumor is enormous, the expansion of the cells, you can get a diagnosis in a real time that you could do 2 or 3 single gene tests. That makes having a whole genome. And we already know from work in childhood leukemias at Great Ormond Street in the new Genomic Medicine Service, where we have childhood tumors live, that they've been able to reprofile treatment just using the genomic report. Now, that's a short read genome. I think there's an intermediary, I'd be interested in John's view on this. I think there's an intermediary stage because ONT and long reads, PacBio is another company that makes long reads. This is still a work in optimization. So it's not ready for accreditation and the level of clinical rigor in the NHS. So for the moment, you do need a short read genome, but I can see a combination of a short and long read genome, perhaps a shallower read, would be an intermediary step. But I do think that there will be a point where there'll be a technology flip. I don't see it yet, but I could see it in perhaps 18 months or 2 years. And we'll be particularly looking at ONT and PacBio in our next phase of work to see what added value. The other thing you get from long read genomes, both PacBio and ONT, is you can read the epigenetics, the methylation status.
Brilliant. Next from Iman01, what are some reasons that some diagnostic yields for specific diseases are lower than desired?
So there could be several reasons for that. First of all, one possibility is that people to enroll against unmet need, they have by definition eliminated the low-hanging fruit. So it may be that disorders in that system are well diagnosed by simpler testing and they don't need a whole genome. So, you know, you can see that there could be a mixed economy. The other reason is that we just haven't done enough research on these disorders to find something. So let me give you a shape of this. When we return the report, sometimes we're able to return 1 clear pathogenic or 2 clear pathogenic variants. Sometimes the architecture of the disease is, we have examples of a rare variant.
I think Sir Mark's frozen
out one version of the protein on 1 chromosome. The common variant reduced. If 3% was still there, they wouldn't have the disease, or they'd have it a lot later in life. But that 3% loss is enough to cause disease. So compound heterozygotes, variants that we don't read well, structural variants with short read technologies, copied regions with pseudogenes that longer read technologies will help us with. So that's why I think the future is a combination of technologies. I also think, to John Todd's point, multi-omics is vital here. I don't yet have the absolute evidence, but alongside the proteomic experiment I described earlier, I'm also measuring 6,000 transcriptomes. And I hope that by integrating genome, transcriptome, and proteome, we'll start to unlock some things we can't see just in the genomic data on its own. And then the other thing is that in many disorders, I'm handing back 1,000 variants, which could be plausible, but I don't have absolute evidence for it, and we need function to characterize that and shrink that. Now, the NHS, to be fair, are never going to have the time to look at that. So it's up to this community to find a way of shrinking that down to a manageable number, and then we can convert it into utility for patients. Roshan, back to you.
So the next question is from Daria Koisa. I hope I pronounced that right. It's about the ethical concerns associated with sequencing neonates. How do we manage the potential ethical concerns of if the uses of genome data in the future become less benevolent than they currently are in schemes such as Generation Genome?
Yeah, so this is something that we're very concerned about, not least for the reasons you mentioned, but also because public and patient trust and confidence is vital to the success of any of these programs. And you risk that at your peril. And if you make a mistake here, and that's what's kept me awake for 8 years, is you make a mistake for the field, so you can set the field back. So we put patients at do, and we've just concluded a very extensive consultation with the public across Britain, the whole of the UK. And you may be interested to know, we haven't yet fully analysed this, but the general findings are the public believe the right moment is here for a newborn programme, because they're persuaded that even a few children could have the possibility of a different life to measure 9 things from a heel prick at birth when we could measure more, and that could usher in an era of potentially genomic prevention. I don't know whether it will, that would have to be tested. Then we should do this by research and be very careful about what we return. Try and make sure we choose things that are fully penetrant, where we have certainty that they're likely to cause disease, and that we don't label people such that they worry about something they'll never get. And then we gradually expand our knowledge base using that newborn resource and research to work out how we should expand that and see if we could usher in an era of genomic prevention together. But also create a platform for innovative therapeutics to come to our country, because we know where these children are, and just do whatever we can to change the landscape of this forever. Because I do believe that genomics has a lot of power that's yet to be unlocked. And I think if we could create such a platform, a hive of innovation, because it's for the commercial entities, it's getting the return on investment. Reasons that the therapies in this space are so expensive is because they're delivered for very few, and therefore they're very expensive, and the trials are difficult to do because you can't find the patients. What if we created a national platform which meant that, subject to people's consent, they could be offered the opportunity? And that's why the recall for research that John was referring to is vital and would be a principle. And here you'd be asking mum and dad to consent in the antenatal period, and you would use the Guthrie spot on the blotting paper to use that for the 9-point test and compare it to a whole genome. That's the argument. And you know, the public were also consulted on what the level and depth of testing should be, and they agreed with the scientific advice. And this was tested with 35 people at the request of the CMO. I chaired a group in 2018, and we suggested that if there was 600 treatable conditions, that should be the focus. So don't boil the ocean, don't make it too wide, don't return things where there's uncertainty. Let's just establish this safely and see if we can change outcomes. And if we can do that, then we can tackle the more difficult situations. And for example, hereditary predisposition to cancer in later life, which you wouldn't tell a child.
Absolutely. Next one is from Holly, actually. So given the hype around microbiomes and their influence on our health and immunity, are there databases of microbiomic information across individuals as well? Or is this something that Genomics England would look towards?
So we're not doing that. I'm not so au fait with the field, but if there aren't, there should be, or there will be. And Holly, you and I were talking earlier before we started, but I share with everybody that we published a paper in Nature. I was just a side author, wasn't me really. And a group in the Netherlands had found in metastatic colon cancer that when they read the genome of the tumor, they also detected the genome of a particular serotype of E. coli. So this is in Nature in January or February last year, 2020. And so they'd found this, and then what they did was they created an organoid outside of the body, effectively a mimic of the colon, and they treated it with this E. coli. And within months, it became cancerous. And so this E. coli was triggering cancer. Now, we validated that finding. We found the same thing, but because it was in a second study, this allowed it to get into Nature. It turns out that E. coli serotype is in several probiotic drinks. So perhaps we should do something about that. But I do agree that the microbiome is going to be much more important. I also have some evidence of importance in upper GI cancers too, but that's unpublished and that's other people's work, so I shouldn't say any more about
it. Yeah, it's very much early days and there's a long way to go.
Yeah.
And then I think this should be our last question from Professor Todd. How does the future of gel fit with our future health?
So I think they're complementary but distinct, but I think I'm hoping that they will work more and more together. To give you an example of how we've been working together, not yet, we are talking to our future health, but as they haven't enrolled, we haven't really had an opportunity to work with them in detail. But with UK Biobank, I've just completed and are about to transfer back 500,000 genotyping arrays that have been 100,000 genome phased genomes imputed. So we phased the genome family members on 76,000 whole genomes. Then we've used that to create a new imputation scaffold with much more rare variant calling, because we have a much more granular picture of the genome. We've run it on a couple of traits, and it looks like it might aid fine mapping, and it looks like it might augment discovery. Clearly, Biobank will have whole genomes at the end of the year, so that's not the issue. To answer your question properly, John, actually, the United Kingdom data assets in genomics need to join up, and there need to be a single seamless front door over the web, like we're speaking now, to access those. So I can seamlessly take my rare disease patients and run a gene burden test using our Future Health 5 million and Biobank. So John and I have done this work. Often we borrow computers at night time from US universities when they're all in bed, and we run our analysis on, in the old days, that's what before we had our own decent compute infrastructure, we used to borrow compute from other universities that would let it be available at night time. So we need to just bring the data either physically together or more likely to federate it. By that, what I mean is to build pipes that connect data centers so you can seamlessly run an analysis across several data assets. And we've got to do that. I think that's Britain's hope of staying ahead of the game. And I think Britain is the world leader in genomics. There's not really much opportunity to do a 100,000 genomes project in the US. Of course, they are doing all of us. That's different from Biobank. And we're doing our Future Health. And so we have programs in disease as well as population cohorts. I think our Future Health is very exciting, but I think these resources must be joined up. That's in the public interest, the patient's interest, and actually it's in everybody here's interest.
Brilliant. And on that note, I'd like to thank you again, Sir Mark. Professor Todd, would you like to respond to that? Or?
I know, I totally agree, Mark. That's the place we need to get to. And you probably need to consider a sabbatical in Oxford. Thanks for everything.
Well, you'll remember, John, I used to inhabit the same premises as you.
Come back. I'm glad I came back.
I might talk to you offline about that at some point. That's a very kind offer. Thank you, John. It means a lot, actually. I'd like to thank you, Roshan and Holly, and all of the Oxford University Precision Medicine for having me here. And I'll say good evening. I'm off to have a beer now. I hope you're doing the same. Take care.